here. So these are the R scripts for each one of these exercises. Now, all of these files, what you need to do is download them into your computer and place them in the same folder. All of them should go into the same folder. And all of these files have the extension .r. That means it's a file that is readable with R and also with RStudio. And in a moment, we're going to open one of these files and we're going to go through the material and solve some of the exercises. So this is the handout, and just read the first page to remember what you did in week 4. And if you review this material, all the formulas are provided here. And we're going to work through these data sets. And we are going to now, we already solved all of these using Excel, of course. Now we're going to solve them using R, and you are going to see how you write all of these scripts for these exercises. Now, the first data set focuses on a study on bumblebees visiting California poppy flowers. And there is a sample containing 7 data points. These are the numbers. This is in number of seconds. And this is the average time, well, the time that each bumblebee took when they arrived to poppy flowers. Now, the exercise is asking to calculate the mean, the variance, the standard deviation, and the standard error. So now we're going to switch to R and solve this exercise. Now, after you download all those files, you have to create a folder in your computer. I suggest creating a folder that you will call a week 9 lab, like the one I have here, the first folder that you see on my desktop. And within this folder, you're going to save all of the files, the R files. So these are the 4 R files that are provided on Canvas, this week's module. As you see, all of them have this extension .r. Now, a very important thing for exercises 1 and 2, they are contained in the same R file because they are, the first one is relatively short, so I just put it in the same file as exercise number 2. Now, the handout is also ideally just save it there so you can always refer to the handout if you need to. Now, in order to work with R, you can just open this file in R, in the base R, and you could potentially start working directly, or you can use RStudio. So what I'm going to do is I do a right click and I'm going to open this file in RStudio. When you do that, RStudio will open in your computer, and typically RStudio has 4 panels, and I'm going to just resize this. The nice thing is that you can hold onto these dividers and you can move them up and down to resize these panels. The panel on the upper left is showing what is called the script. So this is a script, and you can always, if you start a brand new script, you can always use this plus sign on the upper left where you can add a new script. For example, you can click on add a new script and R will create a new tab and it will open a new one. Now, because we have the scripts already saved here, I'm just going to explain in a moment how we do this work. But let me show the other 3 panels. The one on the bottom, on the bottom left, on the lower left is called the console, and the console is basically giving you information about the R version and also the operations that you're going to do. It gives you the R version. Usually the developers, they always choose some name to call, the version they create. So this one is this is the name. And whenever you do an operation, you are going to see something on this lower part of the console. Now, the upper right gives you what is called the R environment. Whenever you open something, it will give you information about that. If it is a file, what type of data are contained in the file. And on the lower right is a panel where you have multiple tabs where you can see, for example, files. These are the files contained in the folder for this week lab. Then we have an empty screen for plots. So whenever we create a plot, the plots will be shown there. We can also look for packages. If we want to add packages to do different operations, those will be listed here. The ones you see on this panel are the ones that I currently have installed in my computer, and You can continue adding more packages depending on the need of the analysis that you're doing. And then you have a couple other things like help. If you are wondering about something in particular, you can type in, for example, let's say standard deviation or mean, or anything, and you are going to get something, you know, arithmetic mean. So anything that you want to know, let's see, you will get information, and then sometimes you get multiple options where you have to, you know, browse until you find the right answer. Now, let's work through the first exercise. We'll just keep recording this part. Whenever you see R scripts that have a numeral, like the ones here in RStudio, numeral lines with a numeral are just notes. You can just add as many numerals as you want. For example, here I can add a new row and just add a lot of numerals. RStudio will color them in green. And that means this is just, this is information that won't be processed by the R program. This is just information for us to remember what we're doing. In this case, I just wrote here 2 rows with numerals, or 2 lines with numerals, and using R in Biology 433 labs, you can add any comments to your own scripts by adding a numeral prior to that. For example, here I say clean up the workspace. This is included by default in my scripts, and that's just to make sure that there are no other operations or things going on under what you see in RStudio. So by running this line of code and the next one, we're cleaning up the line, the workspace. Now, how do you run a line of code? So first of all, this is ready to go. This probably doesn't look familiar to anyone, but this means remove, and we're going to remove anything that is listed there. If I do ls, if I run this line, I will see what is currently contained in my working space or working environment. How do you run the first line of code? In this case, you can click run here, or if you use a Mac, you can do press command and then press return, hold command and then return. If you use a PC, you will press control and you will do control enter or control R. Those 2 options I have seen work in PCs, so you will just have to try on your own which one works for you. The easiest one, of course, if you're doubting, you can always place a cursor anywhere along this line and just click run. There is a button that will allow us to run that line of code. So I'm just, to start, I will just click on run. On the console, you see, I just run this line. The console is letting us know that that's the line we just ran. Now, the next one is line 8. If I press run, it will give us a list of operations or things in our working space, and it will give us 0. And that's what we want to see because we just cleaned up the workspace. Now, let me give a couple of examples before we do the actual exercise. For example, in R, you can do basic operations like, for example, if you want to add 2 numbers, this is essentially like using a calculator. Let's say you want to add 20 plus 30, you click run, and on the console, you see the line of code that you entered, and then you see the result. If you want to multiply, let's say this 20 by 30, and if you want to, let's say, multiply these times 25, let's say, now you can again run that line of code, and now it gives you the answer here. So you can do a lot of operations, and you can, of course, edit things. Like, for example, if I want to call something, for example, AA in uppercase, and I say AA equals 2, you are going to see this operator often. This is called, I think people call it the caret sign, which is basically less than and a hyphen together. This is equivalent to equal or equals to in R. And in this case, we use this caret sign to define something. So for example, if I am calling AA, and I'm going to call this just a value, for example, 150. And if I call something else BB, and let's say I call 2, now you can, if you define those 2, you can run this line, run the next line, and you are just defining those two. Now you can do an operation where you can just add those two. You add AA plus BB. So that's a new operation, and then you can run that. And now it's going to give you the result, so the 152. If you do a different operation, let's say AA times BB, you run that line of code, and now it gives you the answer on the console. And these are the most basic examples, of course, but you can do a number of things that can be potentially more complex. One other thing that is very helpful or very useful in R is using vectors. For example, if I want to define something else like CC, I'm going to define a set of values, and you will use this vector, which is a lowercase c, then you open and close parentheses. By the way, whenever you click on parentheses, RStudio and R will automatically close the parentheses. So, and that means you can add something inside that parentheses. So let's say I add 3 things here, 3 numbers, 2, 3, and 4. Now, if I run that line of code, in the console, you just see exactly that line. Now you can type CC and then run that line of code, and it will give you the numbers. When you do CC 2, 3, 4, so that's a vector. This is called a vector containing 3 elements. The 3 elements are numbers. And now whenever you just call for that vector, like clicking on run here, or just command return or control enter, control R, it will give you those numbers. Let's say now you want to multiply CC times 3. So when you do that, what's going to happen is the same thing as if you were multiplying 3 times each one of these elements. 3 times 2, 3 times 3, 3 times 4. So if I do CC times 3, let's see what happens. So now it's giving us the answer here. So that's the result of multiplying a vector by a number. So this can be very helpful if you want to do, for example, conversions. If you had, let's say, if these are temperatures in Celsius, if you know the formula for converting Celsius to Fahrenheit, you can actually enter that here. Of course, it's a little more complicated than just a single number. And then you can convert those automatically. You can do that with Excel. Of course, nowadays you can do that, just go doing a search in Google, but for a number of operations, you won't have something already built in or ready to go. So you will have to create some of your own operations. So that's just a brief introduction about some of the features of R and how you can do different operations in R. So I'm just going to remove these from the script. This will be, of course, in the video. You can always try it yourself if you watch the video, but I'm going to just keep the script as the original one saved in the on Canvas. Now, with R, we can estimate a number of things like mean, standard deviation, and a lot of other things. But one of the things that is not built in is the standard error. So in order to calculate the standard error, we have to write a function. And in this case, don't worry about writing that on your own. This is already written here. And this is basically, I am defining a function, in this case, standard error equals 2. And then this is a function where we are going to use the formula for the standard error, which is, this is all written in text, but this is basically the square root of the variance of a number divided by the length of the, or actually the variance of a sample divided by the sample size, which this is defined as length of X. So for now, I'm just going to define that function and I'm going to activate that function. So to activate this is just running that line of script. So I click run, you see that it's on the console, so that's ready to go. And we're going to use that in a moment to solve the first exercise. Now, this is a data set that is provided in the handout. The number of visits. I'm creating a vector here, is called visits. So you can, when you create a vector, you can use a single letter, a row, a string of letters, or you can use words that will allow you to remember things. In this case, we're using visits equals 2. And then inside the vector, these are the 7 data points. So these are the number of seconds that it took for bumblebees to visit a poppy flower. And so these are the numbers. So we're going to define the first line of code. So I ran that line of code, and you see on the bottom, what the console is doing is basically mirroring anything that you do on the script in the editor window. Now, a length of visits, that means the length of this vector, and that means how many elements are there in the vector. We know there are 7, we can count them by eye, so there are 7 elements here. We're going to run that line to see what's a result. Length of visits is giving us 7. You see on the lower left on the console, you see 7, so that's the sample size. Now let's estimate the mean of these numbers. So mean of visits, if you run that, that's the mean. Now let's estimate the variance. You click the next line, run that line. So that's the number. You do the next one, standard deviation of visits. So that's a built-in function. Var is variance, SD is standard deviation. Now SE, remember we defined SE, which is the standard error. And now we're going to just run that line of code. And so now you get the solution for exercise 1. And you can, of course, compare these results, these numbers, with the ones you obtained in week 4. You can go back to your handout and see, hopefully you got the same results, and you can just double check those numbers. Okay, now we're going to work with dataset 2. So this is the next exercise. And if you remember, this is from week 4. We did a calculation. We studied data for salmon. There are 2 samples. One is for males, one is for females. Each sample has 30 measurements of the standard length of salmon. So these are 30 measurements of males, and these are 30 measurements of females. And the exercise is asking us to calculate the mean, the variance, the standard deviation, and the standard error. And you will write, you have to write the results. In this case, the main goal is to use the R script and show your work just using the R script. By the way, what do you need to submit at the end of the work? What you need to submit is just the R script at the end of the work that you will be doing, at the end of the week. Now let's switch to RStudio and solve this exercise. Okay, now we're going to close this exercise. This is, remember, actually, sorry, this is the same exercise, same file. So this is dataset 2. I'm going to just make this a little wider here, and I will move this up. Now the dataset for salmon, we have data for males and females. How do we enter the data here? These are, we create a vector, males equals 2, and these are the 30 measurements. So this is a C for vector, and inside the parentheses, we have all those 30 measurements. I'm going to just scroll to the right within the editor console. So you see all those measurements are there. They are separated by commas. You need to add a comma between each one of the measurements. The same for females. We have females equals 2. Remember, it's a less than and a hyphen. And then the vector starts with C parentheses, and you see those 30 measurements. Remember to always close the parentheses. R will automatically add a parenthesis, a closing parenthesis, but make sure you have it. Otherwise, you will get an error. Now we're going to define those 2 vectors. So in this case, we can select the two lines of code at once. So this is one example of running 2 lines of code as a block. And then you can press run, or you can do command return or control enter. So I'm going to just run those 2. And if you see the console, this is basically mirroring what we just did. And now we're going to do something. Let me just add 2 lines of code here. We're going to write just males. Now let's write males and do run that line of code. If you press control enter or command return, you see those numbers here. If you do the same for females, you write females and you do run the line of code. I'm just pressing command and return on my computer. It's the same as pressing run. And now you get those 30 values here. So you see the console is now giving us, all the elements in that vector. A very important thing is that when you look at the console, you are also going to see whenever you run a vector or some operations that have multiple lines or multiple elements, you're going to see what is called an index number on the left side. So you see on the left-hand side, there is 1 inside these brackets. Then you have 15, and then you have 29. These are, I think these are called square brackets. Now, 1 refers to the first element in that vector. Here, 15 is the index number for this element, the first element in this line. So why it's called 15 is because there are 14 on the first line. This is the number 15. And then this goes all the way to, in this case, 28. And this one is 29. So this is just an easy way to see how many samples there are. Here we know, okay, there are 29, there are 30. The same here, we know it's exactly same sample size, so there are 29 and this is 30. Now let's calculate the summary statistics. So this is already written here, range of males. The range is going to give us the minimum and maximum values. And notice here that I have added a comment in the same line of code. I have added a comment, and that's something that you can do for any of your lines of script. You can add a comment in the same line as long as it is not within the embedded in the line of code in the actual script that you will run. So this is separated by just at least 1 space or 2 spaces. And this is just to remind ourselves what we're doing in each one of these lines of code. So here, a minimum and maximum value. So let's run that line of code. On the bottom, you see range. The minimum value is 399. The maximum is 495. And you see that in the console, you see the comment. The comment, having a comment there won't affect the operation at all, because what R does is whenever it sees a numeral, will just skip whatever is written after the numeral. So it's not affecting any of the calculations. Now, length of males, or length, is an operator that allows us to calculate the sample size, or estimate, calculate the sample size. So this is 30. Again, mean is to estimate the mean. So we run that line of code. That's the mean of males. Then the variance, that's a variance. Then the standard deviation. I'm just running the next line of code, and then standard error. And again, remember, standard error was predefined at the beginning. So this function is working and is giving us all those values. Alternatively, I'm just adding this. This is a potential alternative for standard error. You can also estimate the standard error as defining by dividing the standard deviation divided by the square root of the sample size. And if you see here, I'm using the name of the vector, males, and I'm using the sample size, length of males, and I'm estimating the square root. So if we run this line of code, we should expect to get the same value as the one in line 41. So this is line 43. So let's run that line of code. And you see the number that you get is exactly the same. And that's the formula for the standard error. And by the way, this formula is slightly different than the formula that was defined initially. So the initial formula is here. And now we have used an alternative formula. In both cases, it will work. Now let's calculate the summary statistics for females. You see we have written here range of females, length of females, mean, variance, standard deviation. Now that we know that these are written properly and the vector has been defined, we can run that whole chunk of code at once, and we get our results right away. And again, we have this alternative operation for the standard error, and we get, again, the same number here. Now, let's look at the distribution of data. These are additional things that are originally were not required for you to do when you were using Excel, but we can make a histogram with this data. So this is a function for histogram is hist. And then this is the name of the vector. And this is number of breaks is just the number of dividers that I'm adding, how many bins I'm going to separate. And we are calling this histogram S1. So I'm calling this S1. This is arbitrary. I could change this to actually call it hist1, or you can even call it AA, anything. I'm just adding just one number, one letter and a number. And very important, when you create vectors, don't only use numbers. So you need to use a combination of letters and numbers, because if you use a number, that can create potential problems later on. So better to use just pure letters or letters and numbers. Now let's create the first histogram. Click on run. And so that's a histogram, the distribution of data for males. And now you click on the same next line of code. And now we create a histogram for females. And you see automatically is adding the title on this viewer window on the lower right. Now, R has additional operations. These are just to show the additional features that you can use. We are going to overlay the two histograms to compare the distribution of data, the two samples. And an important thing is that you are going to see a few additional lines of code that are, strictly speaking, are not necessary if you use RStudio. But nonetheless, I have included them here because these lines of code could also would be useful if you just run these operations using base R. So in any case, you don't have to remember this, just run this whole chunk of code at once if you want to try this out. But basically, what I'm doing here is I'm telling R, I'm going to create a new plot. I define the outer margins. You can define them, in this case, 0. I don't want to keep any space on the outer margins. And then you plot S1 and S2. You remember S1 is the histogram for males, and S2 is the histogram for females, okay? Now, the next thing here, it looks kind of complicated. It's just a set of values for defining the color and the transparency of the histogram. This is just one way where you can color your histogram. You can just write a color here. Instead of all of this, you can just write in quotes. You can write red if you want to color that whole thing in adjust the color. So let's see, you delete this and red. I can do that. And if I do that, I create a histogram in red. If I wanted to color this, instead of using that whole thing that looks complicated, you can just do, let's say, blue. And I just run that line of code and you see that too. So now I'm just going to, let's see, undo these two things that I just did. And we are now going to plot the same histograms, well, with a transparency and slightly different colors. So let's just run the whole code at once. So all of these. Now I have added, changed the colors, some transparency so that you can see the overlay or the overlap, how the amount of overlap between the two samples. And you can see that overlap is in the darker area over here. We can also, you see that automatically we have defined the number of the labels for the y-axis and the x-axis, and these are actually defined here. I'm just scrolling to the right in these two lines of code. I am adding this y label, number of individuals, x label, body length in millimeters, and the main means the main title, the upper part here, salmon length. This is, and you can add that name and those specifications just to one of those, because then when you plot the second one, those are already predefined. When you plot the second histogram for females, notice that there is one thing that I have added here at the end. It says add equals t. That means add equals true. And that's why the two were plotted in the same thing. Now, what happens if I plotted just one of them? Let's see, I'm just going to redo. This is just an example. You don't have to remember these for the exam, but I'm just showing this example. So if you plotted males, this is data for males. If you forget to add this add equals true, let's say I remove that. Let's see what happens. Now it's only plotting females. So that's why if you want to plot two things or two or more things together, it's better to use this function that says add equals t. You can write t or you can write true, the whole word true there. So that's just the example for salmon. And we're adding a legend too. You can always add more things to your plots. And this is the nice thing of R. You can define additional things and add them to your plot. So I'm adding a legend to show what the colors mean, males, females. Of course, Excel will produce something to some extent similar. I'm actually not very familiar about making histograms with Excel, but you could potentially do somewhat similar graphs with Excel. But up until the previous version of Excel, I don't know how you do histograms in a straightforward way. So that's why I usually prefer to make these plots using R. Now, what is a homework? Homework for this assignment is, if you remember, is very straightforward. The homework assignment is what if you measured one additional individual, one additional male, and one additional female, and included that to your data set. So how would you solve this homework assignment? I'm including a note here. One way to solve this is to copy the vectors defined above and then add the new measurements. And you can try that here. So I'm adding a note here, and I'm going to include that in the example. I think I'm going to include it right away here. Let's see, we're going to copy these two vectors, males and females. And this is what you will do to solve and to finish the homework here. So you will add, you can try that here. So I'm just going to add those here, males and females. So you now, and remember, the handout is asking, what if you measure one additional individual? What you will need to do is just add a comma. I believe is an individual measuring 550 millimeters. Let me just double check that exercise. Yeah, 550 and a female measuring 350. So the male 550 and a female that measures 350. So you see, I just added that additional observation. Now you define those two vectors. And now the easiest way really is just to copy the script for these summary statistics. So you copy that chunk of code, and now you add it under the homework here. So you see, I just added that. And now you can just run the whole thing. For males, you get all those numbers, and notice the only thing, well, all the values will change, really, the mean, the variance, etc. Sample size, we know it's not 30, now it's 31. And that's what you see in the console here. Length is 31. For females, you can do the same. You can scroll up. You can calculate the summary statistics. So you copy that line of code or that whole chunk, and you add it here. And now you calculate all of those. And those are, so you see, you're getting the results right away. If you define the vectors properly, you reapply, you can, that's another nice thing of R. Once you have some scripts written, you can apply them to, if you update your data set, if you collect more data, you can continue running the same scripts. Here. So another way to read the data is from a CSV table, and this CSV table is contained in a website. So I have saved this table in a website. This is a link. So, and we now call this data, and we're going to read what this is equal to, and we have this function that is called read CSV. So read a CSV table, and this is the link. So this table is hosted in a web page. So I'm going to copy that link to see where it is and how it looks. So I'm just copying exactly what's inside the code. And now I'm going to go to that page. And you might not see, sorry, I need to stop sharing. It's kind of annoying because I need to go share that again. It opened in a different format that you won't be able to see if I don't switch the view. So can you see now this table that I have here with two columns? Can somebody confirm, please?
Yes.
Yeah, cool. So you see, I just opened that table, and just to let you know, for the other exercises, all of these data are provided in this page. So I'm going to go just to, let's see. For all of those, you can find them in let's see if this works. I think this is the URL. So I have, for these exercises, I have saved these data sets here. They are already available in a web page. So whenever you run the scripts, those will automatically take you to a web page, and you are going to see, you are going to just connect to the data. You don't have to download the data or even look at them separately from RStudio, but in any case, I wanted to let you know that those data are also available through this page. And that was done on purpose because sharing the actual CSV files and then importing them separately can also be a little bit tricky when you're working with RStudio. So for this exercise, I'm just providing them. You just connect to the CSV and then import and open them in RStudio. The alternative is to save those in your computer and then connect to that folder and then open those. So now we're going to make, again, histograms for the new data. The new data sets, remember, have one additional measurements. And right here on our screen, conveniently, we have the two histograms. You see the data for males and females, so the distribution of data. In these two cases, of course, you see, in general, the sample has, there are 30 observations. It shows, to some extent, it follows the bell-shaped curve to some extent, so there is a normal distribution of data. But now remember, we have added one more measurement that is 550 millimeters, so at the end of this scale on the x-axis, and one that is 350. So let's plot the new histograms. Knowing that we just defined males and females, now they have an additional observation. So let's plot that again. So this is for males, and now this is for males. So you see the new measurement is here, 550. And now for females, now we see this new measurement, 350. Now let's overlay those two again. And I'm just running that whole chunk of code, even the legend. And now you can see the new distribution of data. The two histograms look almost exactly like before, except that now you have one observation for females here and one observation for males here. And now you can imagine how that's going to affect all the parameters that you are estimating, the mean, the standard deviation, etc. So that's the end of this exercise. So once you are done with these first two exercises, this is exercises datasets 1 and 2. Remember, if you add any comments to your scripts, remember to save at the end. So you can click on save. And now if you want to start working on the new exercise, you can just go to File and then Open File, and you add it. The alternative, of course, is to close completely RStudio and then start over again. So I'm going to show you that second alternative, where you will just close RStudio. So you click on File and then just quit session. And that means closing the whole thing after saving. And whenever you quit, after you save the changes that you made in the editor, remember, when you close RStudio and R automatically will ask you, do you want to save anything else? And you say, no, don't save. And that sounds counterintuitive, but that's what you need to do. After you save changes in your script, and when you close the session, don't save. That's important, because otherwise you will be saving every other part that you will be saving what's in the console, what's in the environment, etc. We don't want any of that, because we can always recreate all of that when we run again the script. So don't save, and I'm just